Chapter 4 deals with the flow of genetic information, so here we're going to explore DNA and RNA. So to outline the chapter, we're going to look at the central dogma of biology. We'll look at nucleic acids and the DNA backbone, DNA structure, and in that we'll talk about forces that stabilize the structure and variations in the different forms of DNA. We'll talk about the semi-conservative replication model and the experiments that led to this, melting of DNA and how that's a useful tool in the laboratory, DNA replication and how polymerases are involved in that replication process. We'll talk about RNA and gene expression, and with this we're going to discuss the different types and functions of RNA and then RNA polymerase. We will also discuss the amino acids and codons, and how those codons are going to code for particular things. And then we'll talk briefly about introns and exons in terms of genetic information. So the central dogma is going to illustrate the flow of genetic information. We know that DNA is going to replicate itself. So we have DNA replication that occurs. But DNA is also transcribed. So transcription allows us to go from DNA to RNA. The RNA can then be translated into protein synthesis. So this is the central dogma. We go from DNA to RNA to protein. And again, the DNA itself can undergo that replication process. So thinking about the structure of DNA, DNA is a polymer. And so it's a structure, there's repeating units that are present, these monomer units. And in the DNA polymer, the monomer units are these nucleotides. Now, each nucleotide consists of 3 parts. And these parts include the sugar, the phosphate, and a base. And it's one of 4 different bases that are going to be incorporated here, which we'll see in a second. So we'll see through our study that the sequence of bases will ultimately dictate the amino acid sequence of a protein. So in other words, the sequence of bases represents the information content of our nucleic acid. So that structure is going to be very important. So we do want to differentiate between DNA and RNA. And the main difference here is going to be the structure of that sugar that's present. So it's the sugar component that is going to differentiate between the DNA and RNA. DNA utilizes deoxyribose, while RNA utilizes the ribose. So if you look at these structures, the deoxyribose has 1 fewer OH group, and that's highlighted in red. So deoxyribose, you're missing that oxygen or that OH group, and it's a hydrogen that's in its place. So D or unoxygenated at that position. So there are a lot of similarities between the structures of DNA and RNA. When DNA and RNA both polymerize, they're going to do so through the formation of something called a phosphodiester linkage. So that linkage, it's a negatively charged bridge between the sugars. And we can see that linkage. Here's our bridge between the 2 sugars. And it has that phosphate that's involved in that structure. Now that negative charge is going to be key to the stability of the overall structure, because the charge is going to repel and prevent nucleophilic attack on that phosphate backbone. So it reduces that negative charge is going to help reduce the risk of hydrolysis. And this is in both DNA and RNA. So in the process of that phosphodiester linkage formation, what happens is the 3' hydroxyl group of one sugar moiety becomes esterified to a phosphate group. So we see this, if we're looking at our structure, you have your 3', that 3' hydroxyl, and it forms an ester, or is esterified with that phosphate group. Okay. This in turn is joined to the 5' hydroxyl of the next sugar. So that gives rise to that sugar phosphate backbone. And because of this, because it's the 3' that's that links to that phosphate and then the 5' of the next one, DNA and RNA both have directionality to them. While DNA and RNA do have a lot of similarities in terms of their structures, we do know that there are differences, and the first being the type of sugar that's used in that backbone. But they also have some differences in terms of their bases as well. Now, both DNA and RNA use a combination of purines and pyrimidines. So we have our purines here and our pyrimidines. This is the general structure of each shown here. There's our general purine and our general pyrimidine. Now, both structures are going to use adenine and guanine within them. Same purines are present in the structure. However, when it comes to the pyrimidines, we are going to see that cytosine and thymine are going to be present in DNA, whereas RNA will replace that thymine with uracil. So this is going to be in RNA, but DNA is going to include cytosine and thymine. Now, we did mention that the nucleotide was the building block of DNA and RNA, and that the nucleotides have 3 components: that's sugar, phosphate, and base. But you'll also hear the terminology nucleoside. So what's different between a nucleotide and a nucleoside? Well, it's in the nucleoside, the only portion that's included is the sugar linked to the base. So we're excluding that phosphate here. So here we have our sugar and our base. That's what our nucleoside represents. Sugar plus base. And that sugar is going to be linked, that linkage is our glycosidic linkage, or glycosidic bond. And so again, that nucleotide has the phosphate group that's attached. And while we like to think of these nucleotides in terms of the DNA and RNA structure, there are other common nucleotides that exist. And one of the most common nucleotides in biology is going to be ATP. Not only is ATP the source of the building block for RNA, it's also, we know, involved in energy currency. So it's the major energy currency of the cell. We are going to come back and revisit some of these nucleotides as we move throughout our study, but just be aware that we're not only talking about these nucleotides that are directly related to the polymer of DNA. We're talking about nucleotides that can be used in a variety of different biological applications. Now, when we're talking about DNA and RNA, we do have some nomenclature that we use, and there are some abbreviations that you might come across, so some shorthand structures that will allow us to see more clearly the directionality between that polymer, within that polymer. So you can see some abbreviations written, and we have up here these abbreviations. Sometimes you'll see them written as P, and then the base is PA, PG, PC, PT. So this just is representing that phosphate group that's present between these structures, and it's implied that we have that sugar present as well. Because we do know that that phosphate group is going to be present along that sugar backbone, we can write it with the phosphate out front and then show the sequence of letters, which would represent the bases that are present. Or we can just list the bases and know that we're talking about DNA or RNA, so it implies that sugar phosphate backbone. We can also use a shorthand drawing structure where we're showing that directionality that's present, that 5', 3' directionality. And we can write the base sequences moving in this direction. So the P represents the phosphate group that's present, and then you'll have your sugars that are present. So here's your sugar and the base coming off your sugar. So we have the phosphate, which is esterified to 1, and then your sugar present, and then another phosphate there, and then there's your sugar and another phosphate. And your base is coming off here, show your sequence. So looking deeper into that structure of DNA, hopefully at this point You've heard the story of the structure of DNA, that it was discovered by Watson and Crick. That they actually used X-ray crystallography data that was obtained by Rosalind Franklin. So Franklin and her colleague Wilkins, and you may or may not know that these three men actually obtained that Nobel Prize for the structure of DNA. However, Rosalind Franklin did not, because she had passed away prior to that being awarded, and Nobel Prizes are not awarded posthumously. But it was kind of a collaboration between these individuals that led to the discovery of the overall structure. Of course, the crystallography data being that of Rosalind Franklin's. So from the crystallography data and some model building that Watson and Crick did, we came up with this overall structure of DNA. And the structure of DNA is shown here on the left. And there are some features to this Watson and Crick model. We do have two helical polynucleotide strands that are coiled around a common axis. These are going to be anti-parallel in nature. And they're also going to have right-handed screw sense. So they're right-handed. A second feature is that the sugar phosphate backbone is on the outside and the bases are on the inside. And we see that within the structure. Here's our sugar phosphate backbone out here, and here are our bases that are on the inside. The bases are nearly perpendicular to the axis of that alpha helix. So we can see that in the structure as we're looking. Here are the bases, and we see that they're almost perpendicular to this line that runs through that axis of that helix. The bases are going to be separated by 3.4 angstroms. So 3.4 angstroms between the bases, and the helical structure repeats every 34 angstroms. So every 10 so there are 10.4 bases per turn of that helix. And there's also a 36-degree rotation per base. And then the last feature, the diameter of the helix of this overall structure is 20 angstroms. Okay, and again, we can see this all if we're looking at the structure here on the left. We see this 20 angstrom diameter. We see the 36-degree rotation of those bases within that overall structure. We can see that we have nearly perpendicular base pairs to this overall axis. We can see that the sugar phosphate backbone is on the outside and the bases are on the inside. We also know within this overall structure that the bases are always going to pair a purine with a pyrimidine. So it's always going to be purine, pyrimidine pairing. And we know it's going to be adenine with thymine and guanine with cytosine, so ATGC pairing. And these base pairs will result from hydrogen bonds that occur between the bases. So there are two hydrogen bonds that occur between the adenine and thymine, which we can see here, these two hydrogen bonds. And there are three hydrogen bonds which will pair between the guanine and the cytosine based on the structure. In addition, these bases are going to be stacked on top of one another within the interior of the helix. And that stacking of the base pairs allows attraction of bases for one another through van der Waals forces. So base stacking is stabilized by van der Waals forces, or stacking forces. The bases are also hydrophobic in nature, and so that's going to help them stay within the interior of that helical structure. So Hydrogen bonding helps pair the bases, and then van der Waals interactions help stabilize them within that helical structure. Well, how do we know that A always pairs with T and G always pairs with C? Well, there's some data to support this argument. So what was done was information was obtained about the number of A's, T's, G's, and C's that are present and looking at the ratio between them. So if you look at the ratio between A and T and then G and C, for these numbers, regardless of the organism that you're studying, you see that you get a ratio of approximately 1. Some variation, but approximately 1 between these. Whereas if you instead would take the A and pair it with G, you see that there's greater fluctuation in that number. So it deviates significantly from 1. So this is just data to support this idea that A and T pair together and G and C pair together. So up until this point, we've been looking at that structure of DNA that Watson and Crick came up with, and we've been talking about the general features of that particular form of DNA. And that form is considered to be the B form of DNA. Under physiological conditions, most DNA is going to fall under that B form, or the form that we see in that Watson and Crick model, and that's shown here on the left. But there are other forms of DNA. The A form of DNA, which is shown here on the right, here's our A form of DNA, is a less hydrated form of DNA. A DNA, like B DNA, is going to possess a right-handed double helix, so this is right-handed as well. But there are some structural differences that arise from something called sugar puckering. And we'll see this in a second, but it's based on how some of these groups within the sugar structure go in or out of the plane. So in A DNA, carbon 3 lies out of the plane, and it's a plane that's formed by the other 4 atoms of the ring. So that's going to be the C3 endopuckering, and that actually leads to an 11-degree tilt in the overall structure. So you'll see that we have a tilt in how this helix is forming relative to what we see here. In the B form, we have nearly perpendicular base pairs here, whereas those are shifted in that A form relative to the axis. So in the A form, you have it lying out of the plane. When we're looking in B DNA, carbon 2 lies out of the plane. So just to illustrate this, our A form versus our B form. So remember, in our A form, we have that carbon 3. Here's our carbon 3. It lies out of the plane that's formed by the other 4 atoms of the ring structure. Okay, so it jets out of the plane. And when that happens, we end up shifting, or a tilt, in the overall structure because of endopuckering. In the B form, it's carbon 2 that lies out of the plane. So that's A and B DNA, but we also have a third conformation of DNA, which is Z DNA. This structure, unlike A and B, has a left-handed helix, or screw sense. Okay? So Z DNA is left-handed in terms of the screw sense. Here, the phosphoryl groups in the backbone are in kind of a zigzag structure, which we see with this yellow line here. And so that's where the Z comes from. It's a zigzag structure. Now, the role of zDNA is still being explored. We don't really know much about this particular form, but it has been isolated, and we've identified the overall structure. This is just providing an overview of the different forms. In terms of the rise per base, so the distance between the bases here, the overall diameter of the helix itself, screw sense, is it right-handed or left-handed, what type of glycosidic bond is there, how many base pairs per turn of the helix, and what the pitch is per turn of the helix, and then that tilt from that axis. And so remember, in the B form, that B form was nearly perpendicular to the plane of the axis, well, we're going to see tilts in the A and Z form here. So you want to be able to differentiate between these different forms and know a little bit, at the very least, in terms of the rise, the helical diameter, and the screw sense of these things. Also, that there is tilt relative to that axis that runs through. Now, we tend to like to think of DNA as being this nice long linear structure, but like proteins, DNA can fold on itself and form more elaborate structures. We know that there are some organisms, we know bacteria and archaea, that have DNA that's circular in nature, and so the strand is actually a continuous. Circular doesn't necessarily mean geometric circle. It just means that it's a continuous, there's not an end to it. Okay. So we can see here there's not an end to this overall structure. When we encounter circular DNA, it's possible for the molecule to be twisted and supercoiled into a superhelix. When not coiled, the molecule is said to be relaxed. When coiled, it's in a superhelix state. So superhelical structure, because it's coiled and rolled on itself, this would be considered the more relaxed state here. The implications of coiling, well, it allows DNA to be more compact. Supercoiling may hinder or favor unwinding, which would affect interactions between DNA and other types of molecules that it would encounter or interact with. Now, we've said that DNA is a double-stranded structure, but RNA is a single-stranded structure. And so when we have a single-stranded nucleic acid, it's actually likely that it can fold back on itself and it will form a defined structure overall. So in RNA, we kind of have these common motifs, if you will, one of which is going to be the stem loop. So in this structure with the stem loop, it's created when complementary bases within one strand kind of find each other and come together, and it forms a double-stranded region. So while we know RNA itself is not double-stranded, we can have double-stranded portions within the overall structure. In these structures, sometimes we can have mismatched base pairs, and having base pairs that don't match one another will result in these kind of bulges from the helix. Now that can happen in DNA as well, where you can have mismatched and end up with a bulge. These structures are going to be important for higher order folding of nucleic acids, which will ultimately impact the function of these things. And just to show you that you can have more complex folding based on interactions that occur, here's looking at a structure where we have longer range interactions. So single-stranded nucleic acids can form these more complex interactions between more widely separated bases. 3 or more bases can interact to stabilize these more complex structures, and a lot of times it's the hydrogen bonding that occurs between these longer range bases that will stabilize the overall structure. And again, that's useful for things like compacting and interacting with other substances. So DNA, going back to the double-stranded structure, DNA exhibits what's called semi-conservative replication. And this was first hypothesized and studied in the late 50s, and it was done by Meselson and Stahl. So Meselson and Stahl conducted some experiments, and the idea behind this is that the sequence of bases in one strand determines the sequence in the other strand. So if the strands are separated, you would yield 2 single-stranded templates, and those templates could be used to synthesize new DNA. So as replication takes place, one strand is newly synthesized, and we call that the daughter strand, and the other is passed down, and that's the parent strand. So the daughter's newly synthesized, the parent's going to be passed down. So the experiment that Meselson and Stahl did, it involved labeling parent DNA with what's referred to as heavy nitrogen, or nitrogen 15. It's an isotope of nitrogen. Once the heavy nitrogen was incorporated into the DNA structure, the DNA was placed into normal nitrogen, or nitrogen 14. And the differences between the two forms could be measured as DNA replicated. And because there's a difference in the mass of these, you could use a type of density gradient sedimentation in order to determine the distribution between the nitrogen 14 and the nitrogen 15. So to start this experiment, all the DNA, that parent DNA, had nitrogen 15 incorporated in it. Okay? Then we took that DNA and put it in a medium that had nitrogen 14. So as it replicates and you're moving through generations, you're going to end up with, if you have a parent and a daughter that split, you're going to end up, if this is your parent strand that splits and you're forming daughter strands, so the next generation you have one parent and one daughter, one parent and one daughter. The parent strands would have the original nitrogen 15, and the daughter strand would have the nitrogen 14 in it. And as you move through from one generation to the next, you're going to incorporate more of that nitrogen 14. So you're going to be observing a shift towards that nitrogen 14 that's present. So in the figure, we're looking at the bands between the nitrogen 14 and 15 and how they separate with time. And we see that as we continue through generations, the bands do shift towards that nitrogen 14. And that relative shift can be explained by the fact that it's semi-conservative replication, that parent splits and you form one new daughter, newly synthesized strand, and conserve one parent strand as you're moving through each generation. This is just showing this with DNA structure. There's your original parent, one blue in that next generation, and the red is the daughter strand. Then each of these is going to split. And so you can see if the red represents those nitrogen 14 strands, as you move through, you'd end up with more nitrogen 14. Okay, so we've talked about DNA structure and the different forms, specialty structures that are related to nucleic acids in terms of both double and single-stranded, how you can form those bulges or pair up in different regions. And we've talked about the DNA replication process. Now, one aside here is the idea that a double helix of DNA can be reversibly melted. In this process, heating is going to occur in the presence of acid or base, and is used to ionize the base and disrupt the interactions that take place. Remember that our structure is held together by intermolecular forces, by hydrogen bonds and by van der Waals interactions. And so if we increase the temperature, remember from your Gen Chem days, you can weaken those intermolecular forces, okay, and start to separate these. So we call the process melting because there's some temperature where there's going to be an abrupt change in the conformation. The melting temperature, or TM, melting temperature, is the temperature at which half of the helical structure is lost. So that's what that TM represents. Half of the helical structure is lost. DNA melting via heat does not occur within the cell. Okay, so this isn't something that your body is doing, but this is something that is done in a lab. But what can happen in the cell is we have these specialized enzymes called helicases that can help unzip or unwind that DNA structure. When we're doing these experiments in the lab and we're monitoring this melting, we can monitor it using absorption of light. And we use the absorption of light at 260 nanometers. And this is a concept that's called hypochromism. And in this hypochromism, what happens is the stacked bases, when the DNA structure is present, absorb less UV light than when they're unstacked. So as we start to break apart the structure, we start to see an increase in the relative absorbance at this particular wavelength. Now, melting can be reversed, and that reversal it occurs through a process that's called annealing. It's kind of slow, controlled decreases in that temperature will allow for that. Okay, so going back to this idea of DNA replication, replication in the cell actually uses a lot of different proteins that have to be coordinated. There are more than 20 different proteins that are involved in this replication process. DNA polymerase is one of the very important proteins that are utilized throughout this process. So DNA polymerases are very important. The DNA polymerase catalyzes the addition of a deoxyribonucleic unit onto the growing DNA strand. And it happens one strand at a time, and it happens through a particular process. So the polymerase itself is going to allow us to add that deoxyribonucleotide to a growing strand. And we're adding, we see the addition of one here, and it incorporates into that structure. And then we bring another one in, and it incorporates into that structure, and so on as we move through. But the requirements for this, there are really 4 different requirements as we're moving through. First of all, you have to have the 4 deoxynucleoside triphosphates present, and the enzyme is going to require magnesium in order for this process to occur. So you need all 4 of those deoxynucleosides, so ATP, DGTP, DCTP, and DTTP. You need to have those present, and you need magnesium 2+. That's required by the enzyme, and we'll see more of this when we get into our discussion on enzymes. The new strand is assembled directly on a pre-existing template, so we need that pre-existing template. And we can see that here, there's our pre-existing template on which we're growing. You need a primer to begin the synthesis. So a primer is needed for this DNA polymerase. It's a strand having a 3' hydroxyl group that's bound to the template strand. In addition to the strand occurs by nucleophilic attack by that 3' hydroxyl terminus on the deoxynucleoside triphosphate, and we'll see that here in a second. And then you have to have a mechanism that allows for removal of any mismatched bases. So a mechanism really for repair. And that mechanism allows us to remove mismatched bases. DNA polymerases have activity that allows them to excise incorrect bases. It does it through a separate process. The error rate of DNA replication really is less than 10 to the -8 errors per base pair. So it's a very low rate of error, but when it does happen, we need to be able to fix that or address that. So here we're looking at the addition of phosphate and base to a growing chain. We do have a primer strand that's present, and we said that that primer has to have that 3' hydroxyl there. The primer strand begins with a base paired to the DNA template strand. The base is bonded to the sugar, deoxyribose in this case. That sugar has that 3' hydroxyl group, and that 3' hydroxyl group participates in nucleophilic attack on the phosphorus atom of the sugar, or of the deoxynucleoside triphosphate. So we bring this deoxynucleoside triphosphate in. This 3' hydroxyl is going to nucleophilically attack this. You're going to have these other phosphate groups which go away and leave. And you're going to incorporate this phosphate into that sugar phosphate backbone. So the nucleotide is added, and 2 of those phosphate groups are going to be removed. This process generates another 3' hydroxyl group, which can continue the overall process moving forward. So we can see here that we've incorporated that phosphate within that structure, and we've added that base on there because we brought it in, we've got that hydroxyl group that now can do this again. Now, another little aside within this chapter, while cellular organisms have genes that contain DNA, some viruses will have DNA, but others will use RNA as the genetic material. Viral RNA is copied using RNA polymerase once it's inside the host. And once the RNA has been replicated, the cell is going to die. So the virus results in programmed cell death. And an example of this is the tobacco mosaic virus. But we also have something called a retrovirus, HIV for example, and this is another class of viruses that utilize RNA as the genetic material. Now, they're called retroviruses because the flow of genetic information is from RNA to DNA, rather than that normal DNA to RNA that we've seen in the central dogma. So when a retrovirus enters the cell, the DNA, or rather the RNA, comes in, and the RNA is copied into DNA through reverse transcriptase. So reverse transcriptase takes this RNA from the virus and we really make this DNA-RNA hybrid here, ending up with DNA that's present. Okay. That resulting DNA, after these reverse transcriptase process, that resulting DNA is then incorporated into the chromosomal DNA of the host. And it starts to replicate along with the normal DNA. And so the virus is able to take over that way. Okay, now going back to this DNA, RNA, with gene expression, the information that's within the DNA is really expressed through the production of RNA and proteins. So we said that DNA to RNA to proteins, that's that central dogma, and that DNA goes to RNA through transcription, and then RNA to proteins through translation. Now, there are different types of RNA that are needed in order to decode what's in that DNA structure. And so the main types, these are not the only types, but the main types of DNA we'll look at are mRNA, which is the template for protein synthesis, tRNA, which carries amino acids in an activated form to the ribosome for peptide bond formation. And it's actually the mRNA template which dictates the order that this transfer RNA comes in. There's at least one kind of tRNA for each of the 20 amino acids. And then the last is this rRNA, or ribosomal RNA, which is the major component of ribosomes, and it serves as a catalyst for protein synthesis. There are different types of rRNA as well. Of these types of RNA, these three types, the rRNA is going to be the most abundant, followed by tRNA, and then mRNA is going to be the least abundant here. And again, there are other types of RNA that exist, but these are the main ones that we're going to look at. And we can see the relative abundance here. So rRNA is the highest and mRNA is the lowest. We said that DNA synthesis occurs through DNA polymerase. RNA synthesis occurs using RNA polymerase. RNA is synthesized from a DNA template, and that occurs through that process of transcription. The RNA polymerase is the enzyme that's going to catalyze this overall process. And so here's just a picture of the structure of RNA polymerase. The RNA polymerase is going to require a template. The template is typically a double-stranded DNA. Single-stranded DNA can also be used. However, RNA and RNA DNA hybrids are not effective templates. So the template is going to be double-stranded DNA, or in some cases, single-stranded DNA. But we're not going to use those DNA-RNA hybrids, and we're not going to use RNA for a template for RNA synthesis. We also need what are called activated precursors here. This is similar to what we see with DNA polymerase. So the activated precursors, this is looking at our ribonucleoside triphosphates. Phosphates. This is your ATP, your GTP, your CTP, and in the case of RNA, your UTP. Remember, we don't have that thymine, we have uracil instead. And then it includes or requires a divalent metal ion. And like with the DNA polymerase, it can be magnesium 2+, but it can also be manganese 2+. So there are some similarities to that DNA polymerase. And this metal is required, again, for that protein to function, or the enzyme to function. So with the RNA synthesis, much like DNA synthesis, you have your 5' to 3'. So you're required to have a free 3' hydroxyl group, like we did with DNA. That 3' hydroxyl undergoes that nucleophilic attack on the phosphorus of your ribonucleoside triphosphate that's coming in. And then you're going to add to the growing strand. Now, unlike DNA, the RNA doesn't require that primer to be there. It just is going to start to build on that DNA template. You don't need a primer there. RNA polymerase is also not able to correct mistakes to the same extent that DNA polymerase is. So RNA polymerase, difference is no primer, can't correct those mistakes like DNA polymerase. But the overall process is the same. So then in terms of transcription, this is discussed later on, more in some of your genetics courses. But here, just to point out some of the direction to the process of transcription, RNA polymerase recognizes what's called a promoter site on the DNA template. Okay, so you have your DNA template, and there's some region that's called your promoter site. This is where the RNA polymerase is going to bind, and it determines where the transcription process is going to begin. Now, the regions that service the promoter site will differ when you're looking at prokaryotic versus eukaryotic organisms. Okay. The promoter, so the Tata box in eukaryotic, and this Pribnow box in the prokaryotic organisms. That's your promoter. That's where you're going to have binding, and then determine where it determines where the transcription is going to begin. And transcription will actually begin upstream from where that promoter is. Okay. You can have other regions present, and so some more common, that -35 region in prokaryotic. Sometimes you have a box here that's present in eukaryotic. There are things that can come up here, but the main important thing is that you have this promoter. That's where you're going to bind, and then that promoter indicates where the start of the RNA synthesis is going to be. Okay, tRNA and the codon. So tRNA has an attachment site and a template recognition site. So the tRNA, remember, carries specific amino acids, and that's going to help us form those protein structures. So it carries a specific amino acid in an activated form, which then is esterified to the 3' or 2' hydroxyl group. Specific enzymes will help in this process. The template region is a sequence of 3 bases called an anticodon. Okay, so here's our anticodon, 3 bases here. And this anticodon recognizes a sequence of 3 bases on the mRNA. And the sequence that it recognizes on the mRNA will be the codon. So it recognizes codon on mRNA. Okay. And basically, depending on that codon on the mRNA that really tells us which anticodons are going to be here, and that tells us which amino acid we're going to be bringing in. So with the genetic code, you have 3 nucleotides called that codon, and that's going to encode for an amino acid, and that codon comes from that messenger RNA. When we're talking about the genetic code and that codon, it is non-overlapping. Okay. What that means is if I have, let's say I have a sequence when I'm looking at this, a codon is 3 bases. So it would be these 3, then these 3, then the next 3. Overlapping would be if I had these 3 and then went like that, and the next one came with these 3. That's not what happens. Okay? It's non-overlapping. It has no punctuation. That means I am not going to skip bases in between reading this. It does have directionality, so it's going to go the 5' to 3' end, and it's degenerate. And what that degenerate piece means is that there are, you can have some variation in that codon, and it will still code for a particular amino. And we see that here with this overall structure. So if you're looking at first letter, second letter, and then third letter of that. So the degenerate portion, I can have U, 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 and that would give me this amino acid. I can have U, U, C, and it would give me the same amino acid. So variation in that third position doesn't result in a different amino acid coming in. That helps us kind of limit mistakes that occur. So it's useful to have a degenerate code. So when we're going through and we're encoding our DNA, we do need to think about the different regions that are present. We have regions that are referred to as introns and regions that are referred to as exons. The introns are non-coding regions. Okay, so they're not actually going to go, if this is our DNA structure, our gene, they're not actually going to go towards making that protein. But the exons are those coding regions, and they would. The average human gene has 8 introns, and some have more than 100 introns. Okay, so regions where it doesn't code for anything. Now, if we're going through the transcription process and we're taking this DNA and forming RNA from it, we need to remove those introns from the overall structure. So we actually end up splicing these when we're going through the process of the transcription, the transcription process. So the introns in the primary RNA transcript are going to be removed when we form that messenger RNA, and that's important because we don't want to bring in amino acids based on non-coding regions. Now the exons are important portions because those are coding portions. Many exons encode protein domains. Now it's believed that new proteins arose in evolution by the rearrangement of exons, and this is through exon shuffling. So rearrangement of exons encoding discrete structural elements, binding sites, catalytic sites, etc. We shuffle these things around and you end up with new structures, new domains that form. So exons are shuffled to expand the genetic repertoire. Now we did kind of skip over some of the details of transcription and translation, but we want to look at a nice overview of what's going on with DNA and RNA and the flow of genetic information here. So in this chapter, we really focused on that structure of DNA and the structure of RNA and how we're going to synthesize it, how we discovered the structure, what's required in order to go through the replication process, etc.